This week we begin an analysis that is making sense of taste and some other phenotypes as related to a particular gene called Tas2R38. This issue of The Scientist and articles from it will be available in the electronic reserves for the class. The way that you will determine your genotype for the Tas2R38 gene is called restriction fragment length polymorphism analysis. More commonly, people call it RFLP. There are several different ways to do RFLP. You will use a method based on the analysis of DNA polymerase chain reaction, DNA made by polymerase chain reaction, or PCR. The PCR product of your Tas2R38 gene will then be compared to your phenotype for a couple of different traits. Tas2R38 has been shown to encode a taste receptor protein important for the detection of two chemicals, PTC and PROP. This video outlines the overall method and the individual component methods as well. The first thing you need for RFLP analysis is a piece of DNA. You will analyze DNA from your Tas2R38 gene that's been made in a test tube using the PCR reaction. You sort of can think of PCR as a molecular Xerox machine for DNA, and you can use it to make many copies of a tiny part of DNA found in a larger collection of DNA sequences. The tiny part you will make will be from the Tas2R38 gene, and the larger collection is your entire genomic DNA sequence that you isolated from your cheek cells. A single cheek epithelial cell is shown here. Inside the nucleus is your genomic DNA. It's present as a complex called chromatin, and that's a complex of DNA protein. In addition, floating around inside the cell are lots of other solutes, including ions. So in the lingo of PCR, your genomic DNA is the template DNA, and the PCR product that you make will be your Tas2R38 sequence. To isolate genomic DNA from cells, you need to get the DNA out of the nucleus and separate it from that protein complex. You'll use heat and a strong chelator attached to a little plastic bead to accomplish this. The chelators and the beads are called chelates. Chelators are chemicals that bind very tightly to ions. Boiling or heating your sample causes the cell membranes to melt, those lipids to melt. It also causes the proteins to be denatured, and then they fall off the DNA. And all together you make sort of a cellular soup. Your genomic DNA will be dissolved in that solution of the soup, along with lots of other things, including the ions. The chelax beads will bind to the ions in the soup. We need to get rid of those ions so that the PCR method will work later on. The final step in template isolation is going to be to separate anything that's not dissolved, cell debris, the chelax beads, and separate those things from the DNA in solution, and we'll use a centrifuge to do that. The next step is PCR to make the DNA. PCR needs several different components. It needs template DNA, which we're bringing forward from the isolation. It needs Taq DNA polymerase, magnesium ions, and DNTPs, and those will be present in a mixture called GoTaq green. And it needs primers, forward and reverse primers. All of that gets mixed together in a tube and put into a machine that cycles through 3 different temperatures. First 95 degrees, then 55, and finally 72 degrees Celsius. And it cycles through these temperatures 40 times. This diagram shows how PCR works. There's a video presentation of PCR linked on the Moodle webpage, so please look at that. The end product of the PCR reaction, though, will be a 303 base pair fragment, and it'll be lots of that fragment DNA from the Tas2R38 gene. Now we're ready for the RFLP part. Restriction stands for restriction enzymes. These proteins are found in bacteria and act as a bacterial immune system. Their job in bacteria is to chew up any DNA from an invading bacteria or virus, thereby rendering that invader harmless. Molecular biologists have hijacked these enzymes for use as DNA-specific molecular scissors. Each restriction enzyme has the ability to bind to a particular DNA sequence, and that's called the restriction site. The restriction site for FNU4H1 is shown here. It is GCNGC. Notice that if you read the DNA in the 5' to 3' direction, the top strand is GCNGC, and so is the bottom strand. That kind of sequence that reads the same forward and backwards is called a DNA palindrome. FNU4H1 enzyme binds to that sequence and then cuts the DNA and the sugar phosphate backbone after this first C. The result is one piece of DNA is now cut into two pieces of DNA, or two DNA fragments. How does this relate to genotype determination? Let's take a look at the Tas2R38 coding sequence. The whole sequence is shown here, numbered from the first nucleotide ATG of the ATG to the 101st nucleotide, the final A of the stop codon TGA. The region that we are going to make in our PCR reactions begins at this green sequence, which is where the forward primer binds, and extends through this blue sequence, which is where the reverse primer binds. In humans, there are two main alleles for Tas2R38. An allele is the name for a version of a gene. The three circles here show the places where these three alleles differ. Each of these places is called a polymorphism because there's more than one shape, polymorph, for the nucleotide in the different alleles for that gene. One allele is called the PAV and the other is called the AVI. The name of each allele is based on the amino acids that are present in the protein at three different spots. However, single nucleotide differences, also called polymorphisms, in the triplet codon sequences lead to these different amino acid sequences at each of the three circled positions. In the first position, polymorphism, there's a G in one sequence that leads to an alanine codon, but the other allele has a C in that position that leads to a proline codon. In the second polymorphism, the difference is a T in one leads to a valine, while a C in the other leads to an alanine. In the third polymorphism, an A in one leads to an isoleucine, and a G in the other leads to a valine. If you read those three polymorphisms in a row, you get the AVI allele or the PAV allele. Your genotype determination will actually be based only on the sequence of the second polymorphism, and we'll infer from that if you have a V, you must be an AVI. If you have an A, you must be a PAV. How do we use a restriction enzyme to detect this? So let's look at the sequence. Shown here is the sequence for the FNU4H1 restriction site, so it binds to GC-NGC. This next sequence bit is that second polymorphism of PAV allele, and notice that it forms an FNU4H1 restriction site, GC-TGC. So if you were to mix allele DNA made from this allele with FNU4H1, it would be cut into two pieces of DNA. The 303 base pair piece would now be a 65 base pair and a 238 base pair piece of DNA. If, however, you have DNA from the AVI allele, it has this sequence. Notice that it no longer forms an FNU4H1 restriction site. That second nucleotide isn't a C anymore, it's a T. FNU4H1 will not be able to bind to that, nor will it be able to cut it. So if you have AVI DNA in the tube and you mix it with FNU4H1, nothing happens. It stays a 303 base pair fragment. This results in length polymorphisms associated with each of the two different alleles. In this way, you can use a restriction enzyme to detect single nucleotide differences in DNA. You will use agarose gel electrophoresis to visualize those restriction fragment length polymorphisms. There's a video describing gel electrophoresis on the Moodle site, so check that out. But for now, suffice it to say that gel electrophoresis will allow you to separate a mixture of DNA fragments into separate piles. Each pile results in one band on a gel. So you load them up here, put a current through it, they separate into separate piles. 65 base pair fragment will be smaller than the 238, which will be smaller than the 303 base pair fragment. Keep in mind that as a human, you will have two copies of Tas2R38 gene in each cell. One you inherited from your mother and another from your father. So you'll have the potential to have either two PAVs, two AVIs, or one of each type of DNA in your PCR reaction. And there you have it, how to use RFLP to determine your Tas2R38 genotype.